Hello everyone, welcome back. So we are in the session of overview and integration of cellular metabolism, and here we will discuss neoglucogenesis or gluconeogenesis, where the concepts covered will be the different sites where neoglucogenesis occurs. Definitely we will also discuss what is neoglucogenesis and where neoglucogenesis is happening inside the cell or in different organ. Then what are the different reactions and their respective enzymes, important enzymes or key enzymes related to neoglucogenesis, and also different substrate which are utilized in the process. Now, neoglucogenesis is basically formation of new sugar. The term represents new sugar or new glucose. Now, what happens, as you can see in the this picture, that glucose 6 phosphate can be formed from different molecules. So, this is neoglucogenesis, which is formation of glucose molecule from non-carbohydrate molecules. So, basically what happens, as we have already discussed, that glucose is major fuel for few organs like brain, renal medulla, then also RBCs. They utilizes glucose as their most important fuel. Now, glucose in the form of energy can be stored in glycogen, but what happens, these stores are not always adequate. Suppose we are having prolonged fasting, starvation, or in between meals where the gap is high. In those cases, these glycogen stores are depleted. So, where from the energy will come? So, they will basically energy or the molecule glucose can be synthesized from different non-carbohydrate molecules. So, this process is known as neoglucogenesis, also we call it gluconeogenesis. So, what are the substrates which are utilized as the provider of glucose, like non-carbohydrate substrate? So, important one is lactate. Lactate can form glucose via forming pyruvate. Different glucogenic amino acids, they can enter in TCA cycle, can replenish the intermediates, different intermediates of TCA cycle, and finally can be directed towards formation of glucose. Also, not the whole lipid molecule or fatty acid molecule, but triacyl glycerol, by forming glycerol, can form glucose by entering neoglucogenesis. Now, CO2 fixation mostly does not happen in mammals. This is the pathway which is occurring in plants. So, in animals, what are the substrates like lactate, glucogenic amino acid, glycerol, and also propionyl coenzyme A. Now, where neoglucogenesis occurs? The most important organ, as I already told in my previous class, then that wherever you will see some important metabolic control, you will find liver. So, here also the most important organ for neoglucogenesis is liver, and to lesser extent neoglucogenesis can happen in renal cortex and also the internal epithelial lining present in small intestine. Now, neoglucogenesis sometimes is called just the reversal reaction of glycolysis, where glucose is utilized to form pyruvate, whereas in case of neoglucogenesis, pyruvate is converted to glucose. But definitely if you remember, in glycolysis there are 3 steps which are irreversible. Apart from that, amongst the 10 steps of glycolysis, apart from these 3 irreversible steps, other 7 steps are reversible. So, basically if there is formation of glucose from pyruvate in neoglucogenesis, all the reversal reaction can happen utilizing the same enzyme in the opposite direction. But for those irreversible steps, there are bypass reaction. Let us see what happens. So, in glycolysis, these are our 3 irreversible steps, that is formation of glucose 6-phosphate from glucose, then formation of fructose 1,6-bisphosphate from fructose 6-phosphate, and finally formation of pyruvate from phosphenol pyruvate. Now, these 3 steps while forming glucose from pyruvate in neoglucogenesis should be bypassed with some other enzyme, utilizing some other enzymes following the opposite reaction. So, these 3 enzymes, hexokinase, phosphofructokinase 1 and pyruvate kinase, they are not present in neoglucogenesis, whereas other 7 steps the enzymes remain same. Now, what are the different enzymes for these 3 steps in neoglucogenesis? For formation of pyruvate, for formation of phosphenol pyruvate from pyruvate, these 2 enzymes are utilized. One is pyruvate carboxylase, another is phosphenol pyruvate carboxykinase. So, basically formation of phosphenol pyruvate from pyruvate is a 2-step reaction. Then fructose 6-phosphate formation from fructose 1,6-bisphosphate, there enzyme fructose 1,6-bisphosphatase is used, and finally glucose 6-phosphate is converted to glucose with the enzyme glucose 6-phosphatase. So, these 4 are the key enzymes present in neoglucogenesis. So, basically you can see glycolysis is not a reversible reaction as it is. Similarly, in neoglucogenesis also these 3 steps are not reversible. So, glycolysis and neoglucogenesis, remember, in these 3 steps of reactions they utilize different set of enzyme, and for both of the pathway, glycolysis and neoglucogenesis, these are the steps which are exergonic. Okay. So, let us see what happens while forming phosphenol pyruvate from pyruvate. So, there are 2 sets of enzyme used, one is pyruvate carboxylase, another is phosphenol pyruvate carboxykinase. Now, these are the 2 exergonic steps here, because you can see while forming oxaloacetate from pyruvate, there is formation of there is utilization of 1 ATP, and from oxaloacetate while forming phosphenol pyruvate there is utilization of 1 GTP. So, this is a 2-step reaction, that is formation of phosphenol pyruvate from pyruvate via formation of oxaloacetate, and both of these steps are exergonic, where 1 molecule of ATP here and 1 molecule of GTP here are utilized. Now, formation of phosphenol pyruvate from pyruvate basically involves both cytosol and mitochondria. So, you can see pyruvate, it enters mitochondria. Inside mitochondria, the first enzyme is present, which is pyruvate carboxylase. Pyruvate carboxylase converts pyruvate to oxaloacetate. Now, this oxaloacetate cannot directly go outside mitochondria because this is impermeable to the inner mitochondrial membrane. So, what happens? Oxaloacetate is reduced to form malate. With the help of the enzyme malate dehydrogenase, this is the mitochondrial isoform of malate dehydrogenase present here, and here one molecule of NADH is used and NAD is formed. Next, malate, with the help of one specific transporter, malate alpha ketoglutarate transporter, which we will discuss in malate aspartate shuttle. So, the transporter is malate alpha ketoglutarate transporter. Via this transporter, malate goes outside the outside mitochondrial matrix, reaches cytosol. In cytosol, it utilizes the cytosolic isoform of malate dehydrogenase to reform oxaloacetate, and here again NAD is converted to NADH. Finally, oxaloacetate, with the help of cytosolic isoform of phosphenol pyruvate carboxykinase, it forms PEP. So, here you can see that the formation of phosphenol pyruvate from pyruvate basically involves both cytosol and mitochondria. Now, there is another set of reaction where pyruvate without forming malate can form phosphenol pyruvate. Now, these two routes are important for different precursors. Here, where there is formation of malate, that is required when alanine or pyruvate itself is the source of neoglucogenesis or source of phosphenol pyruvate. Whereas, in case of lactate, where lactate is the precursor for phosphenol pyruvate, it does not involve formation of malate. Rather, what happens? Lactate, with the help of the enzyme lactate dehydrogenase, form pyruvate, and with formation of one molecule of NADH, then pyruvate enters mitochondria. Inside the mitochondria, with pyruvate carboxylase enzyme, it is converted to oxaloacetate. Oxaloacetate, with the help of phosphenol pyruvate carboxykinase, which is the mitochondrial isoform, here it was the cytosolic isoform, and with the help of the mitochondrial isoform of PEP carboxykinase, oxaloacetate is converted to phosphenol pyruvate, which comes outside in the cytosol. Now, why these two types of routes are present? Now, it is because in neoglucogenesis there is utilization of NADH. Now, if you remember, in glycolysis there is formation of NADH from NAD. So, in the reversal reaction in neoglucogenesis there will be formation of NAD from NADH. Now, while utilizing NADH in neoglucogenesis, definitely there is depletion of cytosolic NADH, and NADH concentration in mitochondria is very high with respect to cytosol. So, basically this malate formation is actually the replenishment of NADH. What happens? You can see that here with the utilization of mitochondrial NADH, malate is formed. With the utilization of mitochondrial NADH, malate is formed, whereas this malate, when it comes outside while forming oxaloacetate, it provides NADH in the cytosol. So, basically this is an indirect transport of NADH from mitochondria to cytosol. Now, why it is not required in case of lactate? See, you can see here, while forming pyruvate from lactate, already there is production of NADH. So, basically lactate itself is replenishing the concentration, replenishing the amount of NADH in cytosol by forming pyruvate. So, in case of phosphoenol pyruvate formation from lactate, malate formation is not required. So, this is the physiological modulation of the cell to replenish the concentration of NADH and to maintain the flux of neoglucogenesis. Now, pyruvate carboxylase is the mitochondrial enzyme, and as we discussed, most of the carboxylase utilizes the cofactor biotin. Here, for the allosteric regulation of carboxylase, there is important role of acetyl coenzyme A. So, this is acetyl coenzyme A acts as the positive effector for activation of pyruvate carboxylase. Why? Remember, when there is huge amount of acetyl coenzyme A, so basically those are formed from fatty acid oxidation, which happens in neoglucogenesis. So, more amount of acetyl coenzyme A is formed from breakdown of fatty acid, and that acetyl coenzyme A activates pyruvate carboxylase for production of glucose for supply of energy. Now, this term is important. Neoglucogenesis is energetically expensive but essential. Why? So, we told that there are two exergonic reactions while forming phosphoenol pyruvate from pyruvate. Whereas, if you do remember that while forming pyruvate from phosphoenol pyruvate, there was only one ATP formed. So, for formation of phosphoenol pyruvate, more amount of energy is invested, but it is essential. Why? To maintain the supply of energy in the cells, cells like brain, cells like RBCs, which mostly depend on the fuel glucose. So, neoglucogenesis is essential even if it is energetically expensive. So, we have already discussed these first two enzymes. Now, formation of fructose 6-phosphate from fructose 1,6-bisphosphate, there is hydrolysis of one high energy bond and inorganic phosphate is released in the system. The enzyme used is fructose 1,6-bisphosphatase. Similarly, for formation of glucose from glucose 6-phosphate, again another phosphatase is used, glucose 6-phosphatase, with release of inorganic phosphate in system. So, these are the three steps of glycolysis which are irreversible in neoglucogenesis. For these three steps of reaction, there are different sets of enzymes, and these are the key enzymes where neoglucogenesis is basically regulated. Now, we will discuss a few points about glucogenic amino acids. Now, most of the amino acids which are derived from protein, they finally catabolize to form either pyruvate or different other intermediates of TCA cycle. So, basically they can participate in formation of glucose, and that is why these amino acids are called glucogenic amino acids. Now, glucogenic amino acids here are mentioned with their site of entry, like isoleucine, methionine, threonine, valine. These amino acids, they can form succinyl coenzyme A, an intermediate of TCA cycle. Similarly, fumarate can be formed from phenylalanine and tyrosine. Oxaloacetate can be formed from asparagine and aspartate. Mostly the reactions are transamination or deaminations. Alpha ketoglutarate can be formed from arginine like glutamine, glutamate, histidine, proline, and finally pyruvate can be formed from alanine, cysteine, glycine, serine, threonine. These are the amino acids, they can contribute in forming pyruvate. Now, these are the main amino acids, alanine and glutamine, which actually transport the amino group from extrahepatic tissue to liver with the reaction, set of reaction, transamination or sometimes deamination. Now, basically majority of the amino acids, majority of the neoglucogenic amino acid enter TCA cycle via formation of either pyruvate or alpha ketoglutarate. So, these are the major important glucogenic amino acids present in mammal. So, in this class we have discussed neoglucogenesis, as it is the different reactions of neoglucogenesis. So, neoglucogenesis is basically a multi-step process, ubiquitous in nature, present in all types of cell, where glucose is produced from non-carbohydrate substances like lactate, pyruvate, oxaloacetate or any other intermediate of TCA cycle. In neoglucogenesis, majority of the steps are the reversal of glycolysis, the reversible steps of glycolysis where same sets of enzymes are used, but there are 3 irreversible steps of glycolysis. So, in those cases, those reactions are conducted by different sets of enzyme. The enzymes are pyruvate carboxylase and phosphenol pyruvate carboxykinase, then fructose bisphosphatase and glucose 6 phosphatase. So, this is about neoglucogenesis. In the next class, we will discuss how neoglucogenesis and glycolysis are regulated in coordination. So, basically those are how they are regulated reciprocally, we will discuss. Thank you very much. See you in the next class.